Okay, so I'm going to spend some time. This is a very broad one. They asked me, can you talk about mineral during the transition period? I'm going to focus on something that I really enjoyed, and hopefully there's importance to you. So it's this concept of preventing mineral-related disorders, and I'm going to focus on one that's quite important, which is imbalance in calcium during the transition period. So here's an outline of my presentation. So why do cows develop this transient state of hypocalcemia that's quite common in most cows, particularly as cows produce more colostrum and cows become older? Our assessment of the impact of hypocalcemia on dairy cow health, most of it is based on epidemiology, although there are some small experiments that have tests the impact of inducing hypocalcemia in cows. And then I'm going to spend quite a bit of time on methods to prevent hypocalcemia and what we know at this point that has immediate application to on-farm nutritional management. So here's probably a classic experiment that the group of the National Animal Disease Center did, Jesse Goff, in which they mastectomized old 5 to 7 year old Jersey cows, and they fed a very alkalogenic diet to really induce disturbances in calcium metabolism, a very high potassium diet in a population of animals that typically are susceptible to low blood calcium at calving. So you can clearly see that a major reason why cows develop this transient drop in blood calcium to very low concentrations. These cows here have 1.2 millimolar of calcium in blood. So if not given intravenous calcium, some of them would die, obviously. But you can also see that these old Jersey cows, when you remove the mammary gland and they don't secrete colostrum, then you basically blunt this drop in concentrations in blood calcium around calving. So obviously the mammary gland is a major player in these transient changes in blood calcium in those cows. There's another thing that's quite important. The set point for blood calcium in all mammalian species changes with age. A baby calf would have concentrations of blood calcium of 12 to 13 milligrams per deciliter because of osteogenesis. And as cows get older, or as we get older, our set point becomes less and less. So the ability of the chief cells to respond in the parathyroid gland changes with age of the animal. Okay, so here's just some data from the U.S. industry. This is the last survey that I'm aware of, in which people look at blood samples collected in the first 48 hours after calving, just to look at the prevalence of blood calcium below what's considered adequate in that stage, which would be about 8 milligrams per deciliter or 2 millimolar. And arbitrarily, this was classified as having cows within that subclinically hypocalcemic category, or cows that would be compatible with potentially develop milk fever. And it's very clear that as cows age, particularly from the first to the second lactation, that is sort of the key point in which cows become more susceptible to development of this low blood calcium around calving. Okay, so what is different between this first and second lactation animal? One of the major differences is the ability to secrete calcium into colostrum. So here are experiments in which we've done like 4 or 5 experiments. We did calcium balance. Same thing that Jean-Baptiste was talking about in zinc, copper, manganese. We did with calcium, phosphorus, and magnesium around transition period. And one of the major losses of calcium is colostrum synthesis. And obviously, multiparous cows produce a little bit more colostrum, okay, which leads to a greater irreversible loss of calcium into milk. Okay, obviously, a primiparous cow has a lot more dynamic bone remodeling, okay, which would allow them to bring calcium back into the bloodstream compared to an older animal. But I would attribute a major loss of calcium in colostrum as being one of the culprits for that increased proportion of animals with low blood calcium at calving. Now, there's been some discussion that activation of immune cells because of this pro-inflammatory response that typically takes place around parturition is part of also of the contributing factors for hypocalcemia in dairy cows. So we sat down a few years ago and we look, is this a possibility? What happens if we were to activate all the leukocytes in blood? And that's what we went through the literature to see what happens in terms of if we were to have a model in which all the lymphocytes and neutrophils in blood in bovine were activated, and activation of immune cells results in release of calcium from storage sites within the cell, such as the endoplasmic reticulum. And then there are opening of these calcium channels that bring calcium into the cytoplasm to replenish the intracellular storage sites. And our thought was maybe this uptake by the cells would actually reduce blood ionized calcium concentration. That was the thought process at the time. So we went through all this calculation. You can find this in this paper here that's being published. And to the end, after we did all of this, we concluded that almost none of the calcium, the ionized calcium in blood, which is already not very much. There's about 1.5 grams of ionized calcium in blood of a big cow, maybe 2 grams. So only 0.0007% of that is actually mobilized if we were to activate continuously all the lymphocyte and neutrophil population in blood by some pro-inflammatory signal. So we don't think that activation of immune cells contribute to hypocalcemia in cows because of cells are taking up calcium from the vascular space. But there's this thought process from emergency room work in the literature with sepsis, in which activation of immune system and softening of the tight junctions in the endothelial cells allow leakage of content from blood, including plasma proteins, and that would take content from the vasculature to the interstitium, including calcium. So one of the thought processes is that by inducing inflammation, or an animal that just had calving and went through the painful process, or perhaps she's in the process of developing uterine infection or something like that, or she had retained placenta, there would be leakage of content from the vascular space through the endothelial cells and then get into the interstitial space, and this would lead to a reduction in blood calcium. At least in this particular model here in pigs, when people induce sepsis by giving an LPS challenge, they had animals that were controls, which is the closed circle, animals that receive LPS or endotoxin lipopolysaccharide, which is this here, that induce acutely a reduction in ionized calcium concentration, as you can see. Or they had animals that received the LPS challenge, but they maintain ionized calcium concentrations by giving calcium to those sows or pigs. And what they showed was that there was some movement of calcium from the vasculature, supposedly, because of this reduction here, into the abdominal fluid and into the hepatic tissue, which is illustrated here. So there may be associations with inflammatory stimulus that may lead to leakage and loss of calcium from the vasculature that might further contribute. Obviously, if we have an inflammatory process like this, it may shut down calcium absorption in the gut, perhaps remodeling of the bone by macrophages, osteoclasts may be affected, and that may contribute to a reduction in calcium. And maybe Lance will talk more about this tomorrow with his models of inducing inflammation in cows. But there's also dietary factors that predispose cows to hypocalcemia, and these are probably the most important ones. Diets that induce alkalosis that affects calciotropic hormones. Dietary phosphorus intake prepartum that affects the fibroblast growth factor 23 system that affects calciotropic hormones. And lastly, dietary magnesium concentrations in the diet, which affects the activity of calciotropic hormones, such as the parathyroid hormone. So we'll touch a little bit on each of those. So here's a model that we used a few years ago to induce hypocalcemia in cows using a very specific chelating agent that binds to ionized calcium and the cow excretes that in the urine. So we use dry cows in this particular experiment, and we induce hypocalcemia for about 24 hours. So the product that we use binds to ionized calcium, and the cow excretes that in the urine, mimicking to some extent a cow producing colostrum. Okay, these cows, they went from excreting the urine in the control treatment here of about 200 milligrams to 300 milligrams per day to excreting the urine here about 28 to 30 grams of calcium per day. Okay, so we did that for 24 hours in those cows, and we collect blood at about different times after we stop inducing hypocalcemia. And the question we want to ask is this borderline low blood calcium? Okay, so this is ionized calcium. The total calcium about twice as much. Does that affect the cytosolic calcium concentration in immune cells? So we looked at lymphocytes and neutrophils. Here's the neutrophil data. And as expected, when we induce subclinical hypocalcemia, the ionized calcium concentrations within the cell, which is not surprising, actually is reduced. And obviously, ionized calcium is important for triggering activation of immune cells. Okay? So then we went on to look at, are these cells as capable in vitro of killing bacteria? So we isolate neutrophils from those cows, and we use flow cytometry to look at the ability of those cells, the neutrophils in this particular case, to phagocytize E. coli, pathogenic E. coli from a case of mastitis. So here is at the end of our protocol of inducing hypocalcemia. This is 24 hours later when blood calcium is already reestablished. Here's 72 hours later after we started when blood calcium is completely normal. And you can see that even 2 days later, when you have another population of neutrophils, because of the turnover of those cells in blood is quite fast, they are still not as capable of phagocytizing bacteria relative to the pre-treatment period. And if you look at the ability of having an oxidative reaction to kill the bacteria within the cytoplasm of those neutrophils in the lysosome, it's also reduced. So we think that when cows undergo this process of marginal blood calcium, they may not necessarily be a down cow, as many cows are not. They still suffer with a reduced innate immune response, and that may predispose them to disease. But there are other consequences. So when we induce subclinical hypocalcemia, obviously we're infusing solutions IV. This is a dry matter intake of those cows during the pre-treatment period. We are infusing saline in the control cows. They drop intake by about 2 kilos when we induce subclinical hypocalcemia. And none of these cows went down because we took blood samples every hour to measure ionized calcium to make sure our infusion rate was adequate. They dropped to about 5.6 kilograms per day. There's a major drop in intake. And with that, there's reduction in contractility of the rumen, of the gastrointestinal tract, either because there is less content in the rumen or just because of calcium is important for smooth muscle contractility. But then there are other consequences to that. So although these cows ate less, there is an immediate increase in blood glucose concentrations. Okay? So this is a very typical sign when you have either less release of insulin or you have insulin resistance. And that's exactly what we observed. These cows that we induced hypocalcemia, they had less insulin concentrations in blood, okay, which we attribute in this case to reduced insulin release, although we cannot really prove that concept. Okay? But we know the mechanism of insulin release requires calcium to enter into the cell and cause depolarization of the beta-Langerhans cell so you can release insulin. So when you have hypocalcemia, blood insulin concentrations decrease. And this is going to induce additional lipolysis, which is exactly what happened here. Although the concentrations of free fatty acids are quite low because these are well-fed cows and these are not lactating animals. But my point here is that if you think about this postpartum cow that goes through all the other things that are happening at that period, now if they develop extended period of hypocalcemia, they may have blunt insulin release that can enhance lipolysis at that stage, and they perhaps may have negative effects on the innate immune response. Now, people have worked this out on epidemiological studies. This is one of them from the Cornell group, in which they show the association between different profiles of blood calcium. This is all association now, not cause and effect. And they looked at cows that had no decrease in blood calcium. They had just a slight decrease, but then it went back up immediately. Then they looked at cows that had this transient decrease, but they recover very quickly. Then they had cows that had this chronic decrease in blood calcium, and then cows that had a delayed decrease in blood calcium concentrations. So these are associations. You got to take this with a grain of salt. But what they showed was that the cows that had this drop in blood calcium and an immediate recover in blood calcium concentrations, they are all multiparous cows in this data set, they were the most productive cows, okay? They were the most productive cows. So this did not seem to be detrimental to that cohort of multiparous cows. The cows that were the least productive cows were those that had this decrease and this chronic low blood calcium, which may be associated with other disease. They may have associated disease, clinical disease at that stage, and that's why they never recover calcium quickly postpartum. Okay, so what are the strategies to reduce hypocalcemia? So I'm going to touch on each of them. One is a very old strategy. We've known this for a long, long time, which is to feed very low dietary calcium, below requirements, okay? Such that now cow has to upregulate mechanisms of calcium flux in the body, and that allows them to adapt to that loss of calcium when lactation starts. The other one is to reduce intestinal absorption of calcium and phosphorus by either obviously feeding a very low calcium phosphorus diet, or perhaps using agents that prevent absorption of calcium and phosphorus, primarily absorption of phosphorus. The other one that's probably the most widespread use, at least in the U.S. industry, is alteration in aspartate status by dietary manipulation. And lastly, I'll just show you one piece of data on this, which I think is probably the least effective strategy, which is giving calcium at calving, at least the way we've done in the U.S. industry. Okay, I just want to make a point that although we talk about calcium metabolism in dairy cattle, we know very little about the mechanisms of absorption of calcium in ruminants. We extrapolate data from monogastric, but we really haven't studied that very well. This is a review article by the group in the University of Hanover by Gerhard Breves, in which they pull data from literature, and he looked at the site of calcium absorption according to the intake of calcium by those animals. So there's dairy cows and there's growing steers in this data set. Okay, you can see the reference there. And these animals had duodenal cannula, and they had fecal and duodenal collection and intake of calcium. Okay, so here's the dietary calcium intake in grams per day. Okay, in grams per day. And here is the pre-duodenal absorption of calcium, net absorption of calcium, meaning this is everything that happens before you get to the duodenal cannula. And you can clearly see that when dietary calcium intake is relatively low, they don't find disappearance in this net absorption of calcium, meaning that there's very little pre-duodenal calcium absorption. On the other hand, when dietary calcium is high, there seems to be calcium absorption by supposedly the rumen reticulum, perhaps the abomasum. Okay? So if you look at this data and arbitrarily you draw a line here, it would suggest that for this data set, animals consuming fewer than 100 grams per day, they don't have pre-duodenal calcium absorption. But animals consuming more than 100 grams per day, they would have absorption through perhaps the rumen, omasum, abomasum, before it hits the duodenum. And then they contrast the post-abomasum calcium absorption, okay? So this would be the intestinal calcium absorption relative to the pre-duodenal net calcium absorption. So every time you see secretion here, it means that they found more calcium there than actually the animal consumed, supposedly because there's recycling and maybe endogenous losses, okay? So you can see if there is pre-duodenal calcium absorption, which is here, okay? So pre-duodenal calcium absorption, that is below zero. So basically, there is no ruminal absorption of calcium. Then you observe intestinal absorption of calcium. When there is pre-duodenal calcium absorption, in the cases that we feed a lot of calcium in the diet, calcium is absorbed before the duodenum. They did not find disappearance in calcium in the small intestine. So there's regulatory mechanisms that there's probably some communication through probably calciotropic hormones. Every time we overfeed calcium and there is some perhaps passive diffusion or perhaps active transport in the rumen, that shuts down intestinal absorption of calcium. On the other hand, when diets are marginal in calcium, there's very little supposedly absorption pre-duodenal, then calcium gets absorbed through the classic mechanism of active transport in the small intestine, which I'll show you, which is by a calcium channel called TRPV6. Okay? So, why am I telling you this? Because the classic mechanism of calcium transport in the rumen, okay, are not always properly characterized. So this is the calcium channel. It's called the transient receptor potential vanilloid 6, which is expressed in enterocyte. There is no indication of protein present in the rumen here. So here's the negative control. Here's the rumen tissue. You don't see any band. You can see here in the duodenum. But interestingly, this plasma membrane calcium ATPase, which is the protein that extrudes calcium from the cytoplasm into the plasma membrane outside of the cell into the vasculature, is actually expressed in both tissues. So we do have some of the machinery for active transport of calcium in the rumen, but not all the known machinery that typically is characterized if you were to open a biochemistry book and read about calcium transport. That all comes from mice and rats. And remember, a rat is not a cow, so you don't always get exactly the same type of response. So here's what we know at this point based on literature. So we dug the literature and we went to all RNA sequencing data that we could find in rumen epithelium and intestinal cells. So this is the classic mechanism that you will study in any book. Calcium shows up here in the ionized form, is transferred through this calcium channel, the TRPV6, into the cytoplasm because calcium can induce cell signaling death, binds to a calcium-binding protein that's induced by vitamin D. This transfers calcium to the basolateral membrane, and now we have many proteins that can extrude that calcium to the endothelial cells, to the vasculature. But in the rumen, we have been unable to identify TRPV6, TRPV3, TRPV4 in all the data sets. And there's some indication that maybe calcium proton exchanger is there. Okay. Now, we do have vitamin D receptor in the rumen, although the expression is a lot less than the intestine. Calm-binding D9 is expressed in the rumen, so that would make the case that there's protective mechanisms against increased calcium concentration. And then in the plasma membrane here, the ATPase is present. So there's some indication that calcium transport, active calcium transport mechanisms are present, but some of the other transporters, such as the sodium-calcium exchanger, we haven't been able to find data that have characterized mRNA presence and much less protein. So there's a lot of unknown the calcium transport across the epithelium of the rumen. But there's presumably, transcellular, paracellular transport of calcium through tight junction proteins that get loosened up according to calcium status. Okay, so given this information, what if we feed a very low calcium diet? So this is a classic study by Howard Green when he was a grad student in Wisconsin. And yes, if you feed below requirements, that's what they did. They fed 8 grams per day. Not very easy to accomplish that. You got to have a very special diet. What happens? They have a decrease in blood phosphate in those animals, which may further benefit calcium metabolism, and you'll see why. And you have maintenance of calcium concentrations compared to animals that were fed calcium as limestone in excess. So overfeeding calcium prepartum predisposes cows to hypocalcemia. Okay? So that's the first point. Second point, we don't know a lot about this in cows because we don't have an assay for this protein here called FGF23. But this is the best data that I can find from the German group, in which they characterize mRNA and protein in tissues. Presumably, that's going to influence the concentrations of FGF23. So this is a hormone or a growth factor that regulates blood phosphate and does this by downregulating an enzyme that activates vitamin D3 in the kidney. So it downregulates an enzyme called 1-alpha hydroxylase. The second thing it does, it prevents phosphate reabsorption in the convoluted tubules in the kidney. So the animal dumps phosphorus in the urine. Okay, so every time blood phosphate goes up, FGF23 increases, is secreted by osteocytes to prevent hyperphosphatemia. And this was discovered in the 2000s because humans have polymorphism in genes that control FGF23, and there's some people that have hyperphosphatemia and they don't live very long. There's people that have chronic hypophosphatemia depending upon the genotype. Okay? So they fed a phosphorus-restricted diet, and they look at the expression of FGF23 in sheep. And you can see that if I limit in dietary phosphorus, they decrease blood phosphate. And immediately the bone responds by secreting less FGF23. Presumably, okay, this is the message for the protein synthesis. Whereas in the control diet with high phosphate intake and higher blood phosphorus, now the animal secretes more FGF23 in an attempt to increase urinary loss of phosphate and reduce synthesis of active vitamin D3 calcitriol to prevent gastrointestinal absorption of phosphorus. So then they look at the expression of this enzyme, the CYP24A1, and the expression of FGF23 in bone. This is the enzyme that inactivates active vitamin D3 that makes 25-hydroxy-D3 or 125-hydroxy-D3 into 24-25-dihydroxy-D3. And you can see that as FGF23 increase, okay, remember, high blood phosphate, high FGF23, the enzyme that degrades the active form of vitamin D3 goes up. Okay, again, trying to reduce absorption of phosphorus in the gastrointestinal tract. And then they looked at the concentration of the active hormone for vitamin D3, which is the calcitriol 125, and serum phosphate. As serum phosphate increases, the concentration of the active enzyme goes down. So another mechanism to control phosphorus absorption. But what's the consequence of that? Remember, this steroid hormone here is pretty important to induce calcium transport in the gut. Okay, so that reduces now calcium absorption as well. Okay, and we think that there is a link between high phosphate intake and hypocalcemia that's connected through FGF23, although this needs to be demonstrated in bovine. Okay, but there's some indication. There's studies here in Europe, and here's one study in North America with TMR-based diets in which cows were fed either a control or what they call an experimental diet, which is basically a diet that was supplemented with aluminum silicate, zeolite, that binds calcium and phosphorus, particularly phosphorus. So they reduced blood phosphate concentrations, which was expected. And this reduction in phosphate here, presumably, is the reason for the increased concentrations of calcium after calving in the animals that were fed zeolite before calving. So that's a strategy. If I limit calcium phosphorus intake, I might prevent hypocalcemia because phosphate is linked to calcium metabolism, as I just showed you. So there was also a reduction in blood magnesium, which you typically see every time you improve blood calcium concentration around calving. There's a control mechanism in the nephron. There's a transporter there, the sodium chloride potassium co-transporter system, that if calcium goes up, the kidney now filters more calcium. That competes with magnesium reabsorption. Presumably this drop is because of increased urinary loss of magnesium, although it may be that zeolite also binds some magnesium, although I'm not certain of that. Anyways, but that's a strategy. Now, there's the story of magnesium. So magnesium is an important cofactor here in the adenyl cyclase, which is a second messenger system for one of the calciotropic hormones, parathyroid hormones. So presumably, if I were to feed diets that prevent hypocalcemia, but the animal is deficient in magnesium, so it's not here, like in this example here, the signaling is blunted. Okay, so that's the assumption. I have a diet that prevents hypocalcemia, but I don't feed enough magnesium. My blood magnesium goes down. Now the second messenger system for parathyroid hormone is influenced. People have looked at that not in ruminants, but in other species. So let me show you this data here that makes a good point. So what they did here is, this is a culture of cell culture, okay? And they were looking at bone resorption and parathyroid hormone release in chip cells from the parathyroid gland, okay? So they had two magnesium concentrations, 1 millimolar or very, very low. Obviously, if I were to have 0.1 millimolar of magnesium, this animal would be dead, okay? And what they showed was that with these very low concentrations of magnesium, when you have low blood calcium, you don't stimulate PTH release. Now those chip cells are unable to release parathyroid hormone. So although you sensibilize the calcium-sensing receptors in the cell, there's no release of parathyroid. And that reduces bone resorption. Because when PTH goes up, that binds to receptors in the bone in a particular cell that communicates with macrophages that scavenge bone and brings calcium and magnesium and phosphorus back into the circulation. So probably this concept of low magnesium also affects bone remodeling and the ability to bring calcium back. And then it comes to this concept here that's widespreadly used, at least in the US industry, which is the concept that was put forth by this anesthesiologist from Canada, Peter Stewart, in which cells try to maintain this idea of electron neutrality. If I gain a charge, a positively charged ion, I either lose a positively charged ion or I bring a negatively charged ion, which is something that we erroneously label as anionic salt. Have you ever thought about this? Which salt is anionic? Every salt has a cation and anion. No salt is anionic, but everybody calls that for whatever reason. Okay, so just to make this simple, think of this here, very simplified aspect. Imagine the intestine, we feed sodium chloride, okay? And it dissociates, and let's say that the absorption of sodium chloride is exactly the same, okay? We assume that's 90+%. Both are absorbed, and you bring a positively charged ion, a cation, and an anion. It has almost no effect in blood acid-base balance. It has a very minor effect just because you increase chloride in blood, and the ratio of sodium to chloride in blood is not the same as in sodium chloride. But now, let's say that I feed something such as chloride salts, okay? Ammonium chloride, for example. Let's use that as an example. That ammonium ion is used by microbes in the rumen. Now you have a free chlorine or chloride there. Chloride is absorbed. It's a negatively charged ion. How does that get into the bloodstream? By exchange, for example, with a loss of base excess. So you lose bicarbonate. Now you have less base excess, less bicarbonate. You're going to have more free hydrogen in blood. pH goes down. It's not because the salt is acidic. Okay. Now let me show you another example. This is what we would do prepartum. This is what we would do postpartum. I feed sodium chloride, no effect in blood acid-base balance. But now I feed sodium bicarbonate, for example. That bicarbonate buffer short-chain fatty acid protons becomes water and CO2. The cow absorbs the sodium and does this by exchange with another positively charged ion. In this example, hydrogen gets back into the gastrointestinal tract. Base excess go up, bicarbonate goes up, hydrogen goes down, pH in blood goes up. Okay, so that's how we manipulate that. Okay. Well, if there is one thing that the cow responds immediately, is change in acid-base balance. So here we fed two diets to a group of cows, plus 200 milliequivalents per kilogram, an alkalogenic diet or an acidogenic diet. Immediately, urinary pH begins to decrease, and it reaches the bottom here at about 24 to 36 hours. And as long as she eats the diet, it will stay low because she's compensated to prevent metabolic acidosis. There's no other reason to feed acidogenic diets other than manipulating mineral metabolism because it's bad for cows, okay, to cause acidosis, okay? And blood pH goes down because they cannot compensate completely by hyperventilating, by secreting acid in urine. But every time blood pH goes down, that displaces calcium that's bound to albumin or calcium bound to salts such as calcium lactate, calcium phosphate, and increases ionized calcium. There's an immediate response and then there's a delayed response. So plasma ionized calcium immediately goes up in a matter of a day or two. Okay, so that's exactly what we expect, and we've known this for many, many years. Nothing new. But there's many other mechanisms, and I'll touch on a few of them. There's good data from dogs. There's a beautiful experiment from Spain I'll show you that increases parathyroid hormone secretion independent of blood calcium. I'll show you some data from cattle that if I induce acidosis, tissues respond better to PTH. Okay? When you increase PTH, that upregulates this 1-alpha hydroxylase, that is the enzyme that activates the inactive form of vitamin D3 into the active steroid hormone. This steroid hormone stimulates absorption and transfer of calcium in the gastrointestinal tract. We've done like 4 or 5 experiments that shows apparent digestibility of calcium goes up 20 to 30% in acidogenic diets. But perhaps the most important mechanism is this here that's very seldom spoken about. Every time we have aciduria, pH of the urine goes down. This TRPV5 is highly sensitive to protons. Okay, and the role of this TRPV5 is to reabsorb all the calcium that was filtrated by the glomerulus. So the kidney filters things and then recovers things. That's pretty typical homeostatic mechanism. If I need, it recovers and keeps in the blood. If I don't, it dumps in the urine. This TRPV5 is responsible for recovering calcium and filtrate back into the bloodstream. But what happens is that in the presence of proton, there's amino acid residue there that's very sensitive to pH, and it shuts down the channel. And when it shuts down, the cow pees that calcium out. And now she goes from a cow that loses less than 1 gram of calcium to losing as much as 16 grams of calcium before calving. Guess what happens if you make a cow lose 15, 16 grams of calcium before calving? She upregulates all the other systems, okay, which makes her more capable of dealing with the loss of calcium and colostrum when she calves or she develops disease, so she becomes less likely to develop hypocalcemia. And then there's all this concept of blood-bond remodeling that gets activated by protons. The reduction in blood pH in the bone activates the release of prostaglandin E that works in this rank-Rankel mechanism that communicates with the macrophages in bone. And that brings back buffers into bone, such as carbonate or into blood to buffer the pH. But the consequence of that immobilizes the mineral matter within the bone that releases magnesium, phosphorus, and calcium. So let me show you just some of this. So here's just to illustrate the effect of changing pH in a solution and the affinity of albumin to calcium. If I decrease pH from the physiological range of cattle-fed grasses to when we feed a cetogenic diet, there's displacement of calcium by hydrogen. So immediately you get more ionized calcium just because of that, without any other mechanism. Okay, that's the first thing. But look at this beautiful experiment in dogs. So what these people did is they surgically work with dogs and they put them into a control treatment. They induce metabolic acidosis by infusing hydrochloric acid intravenously, or they cause respiratory acidosis by putting a mask in dogs so they couldn't breathe. Okay? They measure blood calcium baseline for 20 minutes. Okay? And then they infuse EDTA, which is not the best choice, but it works, to cause hypocalcemia, to chelate ionized calcium and dump that into the urine. Okay? But they did this, and they looked at PTH response. So they infuse EDTA, and they measure how much calcium they had to infuse to maintain blood calcium constant. And then they cause hypocalcemia by just infusing EDTA without associated infusion of calcium. Okay, so let's look at this here. So here's the control. Blood pH is at 7.4. This is a carnivore, so it's slightly lower pH than herbivores. Yeah? Then they cause metabolic acidosis, either metabolic or respiratory acidosis. pH goes down, as you expect, very low now, 7.2. They maintain eukcalcemia, okay, which is this here. They maintain eukkalcemia, okay? Then they induce hypocalcemia, and they look at PTH concentrations. Just by inducing acidosis, there's increase in PTH concentrations, irrespective of ionized calcium. Ionized calcium is exactly the same, but PTH concentrations go up, more than double. And then when they cause metabolic acidosis during hypocalcemia, you get a much increased release of PTH by the parathyroid gland. So when animals undergo acidosis, there's greater release or perhaps greater sensitivity of the calcium sensing receptors, and in the chief cells, therefore, there's more PTH release. Okay, so we look at response of cows to giving PTH intravenously every 20 minutes to mimic the pulses of PTH that's released by the parathyroid gland. We fed cows an alkalogenic diet or an acidogenic diet. And then we looked at the response to pumping PTH for about 36 hours in those animals. So they had an increase in ionized calcium concentrations with a negative DCAD, as you can see here. They had an increase in vitamin D3. I don't have time to show you. So if you look at the area under the curve, it's always greater for the acidogenic diet compared to the alkalogenic diet. And this is immediate. Within 3 days of feeding, they become more responsive to parathyroid hormone. Okay? Lastly, I'll show you this data here, which is with sheep, in which they were fed acidogenic diets, yes or no, and they look at calcium transport from the mucosa to the serosa and from the serosa to the mucosa using these Using chambers that shows rumen transport of calcium, okay? So now you would look at that and say, how can we facilitate a passive transport of calcium because we induce acidosis? I think that's not very logical. There's probably an active process. We just have no idea how that works in the rumen. So here is the serosa to mucosa transfer of calcium. There are 2 diets here, and you see no difference. They kill the sheep, they remove the epithelium, mount the Using chambers, and then they look at the flux as if you would go from blood to the lumen of the rumen. And then they look at the flux as going from the lumen of the rumen to blood. And this is the acidogenic diet, the sheep that received the acidogenic diet. Here's the alkalogenic diet. And you can see that the flux is greater, which is this value here, when the sheep received the acidogenic diet, showing that there is greater traffic of calcium from the gastrointestinal lumen in the rumen into the bloodstream, and digestibility goes up. Lastly is this mechanism of shutting down the TRPV5 and increasing calcium excretion in the urine. Okay, so this is the story. Is there a benefit to cows? So we reviewed the literature many years ago, and we looked at risk of disease. And you can see that the risk of retained placenta is less when we feed acidogenic diets compared to alkalogenic diets. The risk of metritis is less. Obviously, if you reduce retained placenta, you expect less metritis. It's unquestionable that you have less milk fever, but you also have less subclinical hypocalcemia. Does the cow respond with more milk? Yes, the multiparous, not the primiparous cows, okay? Whether it's milk or fat-correct milk. We've done 3 experiments with primipares trying to titrate whether there is a sweet spot of acidogenic diet. We haven't found anything. In heifers, we get exactly the same energy-corrected milk, whether we feed the diet as alkalogenic or acidogenic. There's some minor difference in heifer health, but we think heifers probably don't need to be fed those diets. Just control the potassium in the diet. This is an experiment from Stephen LeBlanc's group, shows the same thing. You can see the multiparous cows fed the acidogenic diet, produced more milk. The primiparous didn't. But there were other additional benefits, such as reproductive success. So here is the multiparous cows. you can see the pregnancy to the first AI increase when they were fed the acidogenic diet. No benefit to the heifers. Okay, so we think multiparous cows, they're more prone to hypocalcemia. They benefit from that. I know that my time is almost up. Don't feed a single diet prepartum. There's 3 experiments in literature. They all go in the same direction. In the US, sometimes we want to feed a single diet for convenience. If you use acidogenic salts and if you use zeolite, you definitely should not feed a single diet the entire dry period. So here is an experiment in which we look at reproduction. We fed diets that the only difference was the acid-base balance effect during the entire dry period or only 21 days the classic. You can see the rate of pregnancy was faster in these cows than in these cows. And all 3 experiments that I'm aware of, this is the kind of response. You produce less milk when you feed acidogenic diets for the entire dry period. Because acidosis is bad, yeah? We only do that to alter acid-base balance because of calcium metabolism. People say that products that cause acidosis reduce appetite. They might, but acidosis reduce appetite. So here's an experiment in which we fed a standard diet. There's 5 treatments. I'm only showing you 3 of them. Standard diet. Here's a diet that we induce acidosis. So we include an acidogenic product. And here's a diet that we include the product, but we buffer with sodium sesquicarbonate and potassium carbonate. So then the acid-base balance is exactly the same. So these are dry heifers, about 180 days of gestation. So here's the urine pH, pretty close. Blood pH is close, not quite. And here we reduce blood pH, we reduce bicarbonate, base excess. Intake doesn't change, although we fed the acidogenic product. But when we cause acidosis, intake reduced. Okay, so it's whatever causes acidosis, it can be my product, your product, doesn't matter, it will reduce dry matter intake. Okay, just want to mention this here. Be careful with over-acidifying, okay, particularly if you're using urine pH. That blunts insulin release in the pancreas and reduces insulin sensitivity. This is an experiment that was very complicated, but this is the general conclusion. That affects insulin signaling in cells, and that induces more lipolysis. So we think that people sometimes go overboard with acidification if that's the strategy. Lastly, calcium boluses. There's a lot of literature on that. And if you put that into context and you summarize all these experiments that have been done with thousands of cows, this is what you come up with. There are animals that's very bad to give calcium sulfate, calcium chloride after calving. Primiparous cows and multiparous cows that calve without any problem. The only cohorts that benefit from that are the cows that are older and had a calving problem. Okay? You kill more cows when you give calcium after calving. That induces acidosis, actually. The Cornell studies show more deaths and a more premature culling. So just be careful that sometimes more is not always better. Just to summarize, okay? If you're preventing hypocalcemia, I think it's common sense. Select ingredients that have less of the poison that cause hypocalcemia. Low potassium, low sodium diets. That's what you should do. You should restrict phosphorus intake. Don't feed byproducts that are high in phosphorus, such as canola, meal, distillers' grains. Stay away from those. Make sure you supplement magnesium sufficient. And if you have more soluble source of magnesium, that's ideal. The issue is that there's not a lot of magnesium sulfate. But you can use magnesium chloride if you are trying to cause metabolic acidosis. If you go the route that I showed you more data, yes, choose chloride over sulfur as the acidifying agent. Don't go very low, okay? Don't go too low because that has negative effects. Monitor, use pH. That's a good measure of acidification in the body. Don't feed for too long. If that's not possible, let's say I cannot do this. I don't want to analyze feeds because you have to analyze feeds. You should not use book values. An analysis should be by ICP and not by NIR. NIR, you get a value, and it's always wrong for minerals. Always wrong. So don't use those values, okay? So if that's a strategy you cannot handle, yeah, you have another strategy, which is the use of zeolites to sequester phosphorus. Okay, that still needs analysis of feed. Thank you very much. Most time? Yeah. I don't know the time. Sorry.
Pretty good time for such
checking my things are ringing.
impressive presentation, so informative. Before we go on the break, I want to give an opportunity for one burning question. Opportunity gone. We're going to move into the break. Oh, alert.
Would it matter? As in the US, you have your decades of your lactating cow diets with dry cow diets maybe closer to zero than, for instance, the Netherlands or in Germany, where we have higher nutrition. We then look up the mouse. Do again.
Yeah, so I know what you're going. Like in Netherlands, you have a hard time to get a low potassium diet pre-part, correct? Because you're using grasses that accumulate potassium.
If we need to go towards a low decades to negative, we need to feed an enormous amount of anionic salts. So what's your...
I think very simple. Here's my take on this. You have the poison, that's potassium sodium. You have the antidote, which is not a free antidote, costs money, and it can have negative effects, which is the acidogenic salts or products. What you have to do is make as much effort as you can to take away the poison. Okay? Select forage that are low. It's not always possible. And then add as little as you can of this. Now, if you tell me my DCAD before I add anything is plus 300 milliequivalents, that's trouble. It's very hard to deal with that. You're going to have to overfeed this. That's going to be expensive, and you're going to have a lot more variability in response. That's been my experience. So maybe in those cases that, hey, my diets are so high in potassium, maybe the strategy would be to sequester phosphorus or something similar. Yeah, that's a tough one.